Good morning, students, staff, and colleagues. I'm Dr. Ethel Gordon from the Department of Biology. It is my pleasure to introduce Dr. Kevin Esvelt as the inaugural speaker for this week's Earth Day program. Dr. Esvelt would have addressed you last month during the annual Darwin Festival had it not been for inclement weather. Dr. Esvelt received his Bachelor in Chemistry and Biology from Harvard Med College in Claremont, California, and he received his PhD from Harvard University. Dr. Esvelt is an Associate Professor of Media Arts and Sciences at MIT and Director of the Sculpting Evolution Group. His lab is working to develop technologies that allow for local alteration of organisms. They also are developing systems that would allow genetic changes in the wild to be overwritten or reversed. An outspoken advocate of sharing research plans to accelerate discovery and improve safety, Dr. Esvelt's lab seeks to promote beneficial advances while safeguarding biotechnology against mistrust and misuse. He is here to describe some of his recent work. His talk is entitled, Should we Engineer Ecosystems? Please welcome Dr. Kevin Esvelt.
Thank you for the very kind invitation and introduction, and as well for the oddly appropriate mouse microphone. I love it. So I thought I would, in honor of Earth Week, give a talk focused on this question of what should we do with our newfound power? So the question might be restated as, why engineer living things to begin with? Not necessarily out there in the environment, but just across the board. Curiosity? Is it wanting to understand the world and how other creatures experience it? Understand how they work? Understand how they came to be, how we came to be? But to draw on Darwin Day, Darwin's bulldog, Thomas Huxley, had a different idea. So responding in part to the development of social Darwinism, the movement that says that we should reconfigure society to be more like natural selection in the wild, Huxley said, this is nonsense. Let us understand once and for all the ethical progress of society depends not on imitating the cosmic process like social Darwinism, still less in running away from it, but in combating it. That is, he is saying, evolution happens out there, and we don't approve, so we should do something about it. And he says, it may seem an audacious proposal, thus, to pit the microcosm against the macrocosm, and to set man to subdue nature to his higher ends. But I venture to think that the great intellectual difference between the ancient times with which we have been occupied and our day lies in the solid foundation we have acquired for the hope that such an enterprise may meet with a certain measure of success. Now, it's been 131 years since he wrote that, and we were in fact quite far from doing anything in his day. But we do in fact have a solid measure of foundation for success. So what would we use it on if we decided to? I submit Cochleo hominivora has to be one of the best named creatures out there. In South America, it's commonly called the bichera, or gusano barrendador del ganado. We call it the New World screwworm. I submit that, at least by back of the envelope, eradicating this organism from the wild, removing every last screwworm botfly, would relieve more animal suffering integrated over the future than any other single possible human action. Why is that? Well, this organism lays its eggs, its primary meiasis, it lays its eggs in open wounds, anything as small as a tick bite. And the larvae then drill their way in. That's why they're screw-shaped. That's why it's called the screwworm. And they devour the organism, the healthy tissue, not just diseased. And they culture microbes that emit volatiles that summon in new gravid females to lay more eggs. It solely preys upon mammals and birds, which we know can suffer, warm-blooded things. and it often ends with the host being devoured alive by flesh-eating maggots. So Darwin lost his faith over the Ichneumonidae, which just do this to insects. This is mammals and birds. We're much more confident that they suffer. And by back of the envelope, based on habitat density in South America, it sure looks like this happens to probably something on the order of 1 billion mammals and birds every year. Typical lifetime of an insect species is several million years. When you do the math, this is 10 to the 15th mammals and birds being devoured alive by flesh-eating maggots. For comparison, factory farming is about 10 to the 11th animals per year. So if we were to continue it 100 years before we stop, then that would be 10 to the 13th. So stopping this is probably more important than ending factory farming immediately, unless it is 100 times worse to be a broiler hen than it is to be devoured alive by flesh-eating maggots. It's also a horrific burden on some of the poorest people in South America. Causes tremendous amount of economic damage to herders in particular, because they have to constantly treat their animals for infestations. So I went down to Uruguay, and they have now launched a project to build a gene drive system in these organisms. So CRISPR-based gene drive basically says, let's introduce the CRISPR system and a desired change. Introduce this into the reproductive cells. CRISPR cuts and introduces itself into the genome. But then it's in there, it cuts the other chromosome, copies itself over, such that now when this mates with a with a wild-type organism, then in each generation in the reproductive cells, you get CRISPR copying such that it's always inherited. So generation after generation, it spreads. So the net effect then is me having to drop out of the presentation and to deal with that video. So you end up with the whole population, in principle, being engineered, if this works well. This works great in the laboratory. It probably works great in the wild, but thankfully, no one has actually released them yet, because it will spread to all populations connected by gene flow. So Huxley was pretty optimistic back then. Now we really can engineer entire species. This probably could get rid of the New World screwworm, because we already eradicated it from an entire continent, North America, just by irradiating the larvae enough so that they became sterile. They still developed, and then releasing 100 times as many sterile screwworms as there were wild screwworm flies. And we did this to push them up through Florida, west, down through Texas to the Mexican border. US then made agreements with Central American countries and pushed them south. And so that now, our tax dollars go to releasing 10 million irradiation sterilized screwworm flies every week in a living wall across Panama to prevent them from reinvading North America. There's far too many in South America to get rid of them that way. You really don't want to do sterile insect technique releases over the Amazon. But gene drive could do it. So the problem is, who gets to decide? What should we do and how? Probably the most morally important application is to get rid of malaria and then schistosomiasis. But that probably shouldn't be the model of how we decide whether, when, and how to edit species. Just because it's really hard to deal with power dynamics and complications and issues of education and equity in places like Africa that suffer from those diseases. So the screwworm is an interesting case because Uruguay is a high-income country. They've been engaging in community discussions about whether or not to do this, and of course they would need to involve the rest of the Mercosur countries. But we thought, well, at least I thought, since CRISPR-based gene drive is my fault, we needed a better guide to engineering ecosystems with communities, with community input to help us decide when to do this. So I thought of a few general rules for engineering complex ecosystems, which is, number one is to be humble. Because our power, I submit, comes from reductionism. It's simplifying the system, understanding the pieces extremely well and how they fit together, such that we can engineer a robust machine, macroscopic or microscopic, that will function across a wide variety of environments. But to get that power, we simplified away all of the complexity, which means we're much better at building things that change systems than we are at understanding complex systems made of many different pieces. So this demands some amount of humility in deciding, are we ready to tinker with that system? So if you are going to tackle something, you should try to make the smallest possible change that you think has a good chance of solving the problem, just because the most minimal change is the least likely to break something important. You should always test it by starting very small and local, small scale. Don't do something large. Don't use a self-propagating CRISPR gene drive. That is not suitable for field trials ever. And then scale up only if the results look good in that ecosystem and in fact all relevant ecosystems. And always, always seek advice from and defer to others who know more. Because this is not nuclear physics. If you're running a high energy super collider, there's probably no need to go to the local community and say, hey, what do you think's gonna happen? Can you give us any advice on what we should do? But if you're engineering ecosystem, people live out there and they know a lot and may have noticed things that you haven't noticed, and what we have found is that this is in fact true. So where do you start? Because, like it or not, a lot of people are understandably leery of our tinkering with the stuff of life. A lot of people really don't like genetically modified foods. A lot of people mistrust vaccines. How can we avoid mistakes and backlash? How can we build trust? How can we earn that trust? And I think the key is you gotta address a problem obvious to everyone, because honestly, tomatoes that go bad in the supermarket a little faster than you might like is not exactly a great clarion call to begin tinkering with the stuff of life. You should openly share your proposals before your experiments begin. Note, this is not how we normally incentivize folks to do science. Normally, if you tell people your brilliant idea, then someone else with more money and hands will get it done first, publish it, scoop you, and they get all the credit and you get none. This is arguably fine for biomedical drugs, because if my lab develops a drug and your doctor recommends it to you, you can say no. You can choose not to be affected. You can opt out of being affected by our research. But if we develop an eco-technology intended to change the environment you live in, even if it goes up for a vote in your community, up or down, and you get outvoted, you're getting affected no matter what. So if we developed that one in secret, we denied you a voice in decisions intended to affect you, but you won't be able to opt out of. So you need to actively invite concerns and community guidance openly before you do anything, and arrange for independent assessment of how it's going, because of course, if you've developed the technology, you are the single most biased person in the world when it comes to deciding whether or not it should actually be used. So how to test whether any of this is right? This all sounds good in theory, but a lot of things sound good in theory and don't actually work in the real world. How do we test this? And if it works, how do we actually change those scientific incentives to something that might be more helpful? Because I don't think anyone rationally engineering the scientific ecosystem from scratch would set up the incentives for all of us to keep our best ideas a secret from one another as long as possible. So we decided that living here in New England, a possibly relevant, sufficiently obvious problem is Lyme disease. Now, I'm from the Pacific Northwest, where we don't have horrific blood-sucking monsters lurking in the underbrush all the time, ready to latch on and feed on your blood and give you nasty diseases. So I found moving here a little traumatizing in that sense. I had to be a little bit more cautious when it comes to hiking about. But it is true that one could accurately claim that in the Northeast we have no traditional natural disasters. We don't really get serious hurricanes. We don't have earthquakes or really serious tornadoes, at least not near the coast. Our natural disaster is Lyme disease. It's the most common vector-borne infectious disease in the United States by a long shot. And most cases go undiagnosed. It's hardly malaria, but it ruins lives long term. So we thought, is this something that you could plausibly use an ecotechnology to fix? So unlike Europe, which also has a Lyme disease problem, we have one primary reservoir. It's not the only reservoir, but the white-footed mouse, Paramyscus leucopus, is the primary reservoir. And when it gets bitten by a tick gets infected. When an infected tick bites it, the mouse gets infected. So mainly you just have this cycle between the two of them, and then most other species are secondary. The problem comes when a tick that has just bitten a mouse and gotten infected then bites a person and infects them. And we made this problem worse because we already engineered the local environment to maximize the number of infected ticks. Because, of course, we love our trees. We love living amidst trees. The Northeast is more forested than it's probably ever been. But we've carved it up with roads and houses. So we've maximized the forest perimeter. And as it turns out, the mice do best at the edges of woodlands. Not in the open, not in the dense woodlands, but at the edges. So we just turn the habitat to maximize the number of the primary reservoir. We also got rid of most of the wolves, which allowed the deer population to explode. The deer also prefer forest edges. So we have many, many more deer. And I know some people, having been influenced by Bambi, see deer as these majestic creatures, and they love seeing them in their, well, maybe not in their gardens, but in their backyards. But I can't help but look at a deer and think, that's 1 million ticks in the next generation. So we maximize the number of deer, and therefore maximize the number of ticks. And then we maximize the number of the reservoir that transmits Lyme disease more effectively than any other reservoir. So we pretty much maximize the number of infected ticks in the environment inadvertently. So you can say this is why we have such a Lyme disease problem today, whereas we really didn't, say, 80 years ago. How do we fix it? Well, one possible idea is to engineer the mice so that they can't be infected. Because then you break the cycle. Fewer infected ticks means fewer infected normal mice or other reservoirs in the next generation, and eventually it spirals down and down and down to a much lower level. Wouldn't completely prevent Lyme disease, but it would be much more like it was historically by essentially removing the fuel in this fire. So how could we do that? Well, the idea we had back in 2015, 2016 was, look, you can obviously vaccinate for Lyme disease through Borrelia burgdorferi, OSPE, works perfectly well. Or you can just infect a mouse. It will develop immunity. Some of that immunity is mediated by antibodies. Why not go in and find the relevant antibody encoding gene and move it into the germline of the white-footed mouse such that it's born expressing neutralizing antibodies against Borrelia burgdorferi, the causative agent of Lyme. If all of the mice were born with protective immunity, that would do it. This seemed the sort of thing that we could do. Folks have engineered various animals to secrete antibodies in their milk. Why wouldn't it work if every cell, or even just the liver cells, say, were secreting antibodies into the serum all the time? But again, we wanted to go to communities before we did anything in the lab. So we didn't test anything like that. We actually said, look, this is probably going to work, or something like this is going to work. We can make this work. We don't know exactly how, but it seems very doable. So let's go to communities and ask. So we thought, well, okay, if you're going to change the environment with organisms that can move, you should pick somewhere where they can't move very far. So the obvious choice was the islands, Nantucket and Martha's Vineyard. So Nantucket is a little bit more organized than the Vineyard, for those of you who've ever been there. And so they said, yes, come out, speak at the Board of Health meeting. And so I did. And these are the original slides. And the point that I said was, look, we're not doing this because Lyme disease is the worst problem ever. we can walk away if you don't want this. This is primarily about pioneering a new way of doing community-guided technology development intended to set the expectations for how eco-technologies are developed in the future. And the idea here is that extremely well-off, well-educated communities with a tradition of New England town hall democracy are perhaps best positioned to actually make this work. In other words, if you can't get it to work in an English-speaking, well-educated New England town hall democracy, you probably can't get it working anywhere. So we should figure out the basics here, where it's in the easiest environment, and then that can hopefully set expectations elsewhere. So we emphasize that this is only a concept. It will only move forwards if embraced by the community. It would not be available anytime soon. Lots of people have the idea that technology advances like this, and while that is arguably true in AI right now, it is not true in many other areas. And so we said these are the key questions. Do you want us to do this? Yes or no? Otherwise we walk away. We haven't done anything, we haven't raised any money. What are the criteria for release? What do you want us to do? Immunism versus Lyme? Should we try to engineer the mice to kill the ticks, which we think we figured out a way that we could do that, although we haven't actually done it yet? Or both? Where would you want the first field trials to be held? Who should do the monitoring assessment? Because it can't be us. And we went back again and again and again and again and again. And we tried to get a lot of press to set an example, which worked very well until the pandemic made everyone much more concerned about a different kind of exponentially spreading biological construct. So in the meantime, we decided to figure out, okay, can we actually do this? So we were able to raise some support, both philanthropically and from NIH. But as expected, with 25 million years between your standard lab mice and white-footed mice, they're actually more closely related to hamsters. And it turns out that reproductively, they're nothing like lab mice. So engineering lab mice is now very standard, very straightforward. Turns out if you encode antibodies in the germline, at least it looks like the mice end up resistant, or lab mice. But white-footed mice, engineering them at all, proved to be extremely challenging. So I'm primarily an engineer. We do science and engineering, but my very strong preference is applied science to engineering, not basic science. So I'm frankly extremely miffed that we had to work out the reproductive biology of white-footed mice almost from scratch in order to engineer them. Fortunately, I have very talented folks in the group, because what we ultimately needed to do was figure out when in heck these mice, the females, are in estrus. Because no one knew what the cycle was. There was no mating plugs. You couldn't tell when they had mated. You couldn't tell when they had mated productively. The actual colony expansion rate was terrible, because when you just mix the males and females, the females don't get pregnant very often, oddly enough. And it was just a total mess. You can't do pseudo-pregnancy, you couldn't do anything. And also you can't just give the females hormones and have them produce a lot of eggs, which is our standard practice for lab mouse engineering. Peromyscus, they'll produce the eggs. It's just all the extra ones, they're not viable. So you get a lot of them on paper, and like the number is high, but then when you actually try to fertilize them and implant them, So in order to solve this, we figured out eventually that the females exhibit a unique pattern of movement that is temporal. So on the day when they're in estrus, they remain active much longer. And this is just the females and only on the day when they're in estrus. So we set up these Raspberry Pi-based camera systems. So you can see one here between all of these cages. And we wrote some software, so now we have our mouse app that keeps track of every mouse in the facility is on camera, at least all the females, so that we can actually know what they're up to. And so here you can see now, these are days from mating, so these are cycling females. And you can see every 4 days, boom, boom, boom. This is the hours from lights off. The activity pattern, they extends longer. And then up until mating, you actually add a male on the day of estrus, and then they will reliably reproduce. And then we can do all the embryology and see how, yes, indeed, there is very different developmental timelines of the embryos and the like. So when we had to test out, okay, putting them in which horn of the uterus is actually worthwhile, or can you do both? And we did eventually get some engineered pups. So now we can, in fact, engineer the germline of white-footed mice. This is one of those cases where the planning fallacy always applies. Turns out it took us better part of 7 years to figure out how to engineer the mice at all, because there was nothing and they were just not very similar. So now the next question is, how many antibodies do we put in? Because we want this to be evolutionarily stable resistance. So one of our collaborators has now isolated Paramyscus leucopus antibodies against Borrelia ospae that bind to different epitopes. And they all appear to be neutralizing. So we want to express, ideally, at least 4 antibodies, so as to ensure that Borrelia doesn't evolve to escape. And then the other question is, do we want to add something to localize them? So the communities were very clear on one thing. Do whatever you can, but do not add genes from any non-mouse species. And not even a non-mouse, any non-Paramyscus leucopus. That is, you're allowed to go in and rearrange genes as much as you want, if it's already been in there. But don't add anything new. This is an opinion that is held almost exclusively by people who did not have a formal education in STEM. The community divides almost perfectly. People with a STEM background do not care where the genes came from, they care what they do. People without a STEM background say, no, just as a general rule of thumb, don't go mucking with moving genes across species boundaries. We're worried that things might go wrong, which is a pretty good heuristic, actually, when you think about how much can I get away with engineering an organism using only its genes versus nature's entire toolbox. You really are tying my hands behind my back if you're saying only that species. So it's actually pretty good for this, and since we can solve the problem, fine. But that does mean we can't use CRISPR-based gene drive, which is fine. We didn't want to do that immediately anyway, and in fact, that's the one thing we told the community is, no, we won't do that for you. But here is something that we could do. And so this is what's called underdominance. And the idea here is if you engineer mice to express these protective antibodies, looks like they only need 1 copy of the antibody expressing locus to be resistant, as you would expect. But if you don't want those mice to spread outside the area of interest, if you wanted, say, if Salem wanted them, but Ipswich didn't, or something like that, or Peabody didn't, how do you keep the genes where the people who want them actually live? How do you keep them from crossing the border? And the answer is you give the genes the right to vote. You set up a majority wins dynamic that favors whichever genes are in the majority. So this is what happens in underdominance, where when you have something like a reciprocal translocation between 2 chromosomes, whereby if you need to inherit either 2 translocated copies or 2 original copies, because if you inherit the wrong mixture, if you're 3 in 1 of any combination, you're toast. And so what this does is it says, if you mate with the other kind, half your grandchildren will die. Because that's what happens to the children of hybrids. That's a huge fitness penalty. So in effect, what you do is you favor whichever allele is in the majority. And this would then lead to effective separation between the mouse populations in different communities. So we're working on doing that too. We have to drop in Cre recombinase and in order to get the translocation to work, so looks like we're going to try. So in summary, the communities set up steering committees to guide research and development. We suggested that they include 1 dedicated skeptic, someone who actually opposes releasing the mice and is known to do that publicly, but still supports the project because they like the way that we are going about it. That is, a lot of people support the process. They really like the idea of community-guided eco-technology development. They recognize they're probably going to lose that vote, but they like the idea of setting the standard that this is how it's done to capture community voices early. Again, they prefer cisgenic mice, so no genes across species, and they have actually suggested some things that might have gone wrong that we hadn't thought of. Primarily for if we had made mice that actually kill the ticks and how they're released. So now we can get started on this. So these are our colleagues from various universities and all of the folks in my lab who actually did the work. So that was easy mode. So far it seems to be going pretty well, albeit on the technical side. But what do you do when the government, not you, wants to use the technology? That's a little bit more complicated. Can you do anything? I mean, you have a voice. You're known to have invented it. What do you do? When the relevant communities are more challenging to work with than New Englanders, and when those communities possess a lot of relevant ecological knowledge, how do you understand it? So these were all questions that we faced because the government of New Zealand decided that they wanted to get rid of all invasive mammals by 2050, using whatever technology was acquired. Well, what technologies do you think they started looking at? So we thought maybe perhaps we should go and see what the Maori thought about this and whether they thought it was a good idea. Among other things, the Treaty of Waitangi gives them sovereignty over the natural environment in Aotearoa, not the government. But also, they have among the most well-conserved bodies of traditional knowledge of their environment of any indigenous community in the world. So we thought we should find out before tackling these mice, or these mice and rats particularly. The rats is what they're most interested in. Is this a good idea? How should one go about it? Is it okay for scientists to begin doing this? Should the government be pursuing it or not? And what would they want? And we wanted to ensure that there was Maori and local guidance of these technologies. So the caveat slide was that none of this exists in mammals. Don't expect this anytime soon. And for our part, we're interested in suppressing rodents for other reasons. They're pests around here. They're also invasive here, actually. But they've been here for long enough that I'm not sure what would happen if we completely got rid of them. That doesn't seem very feasible. But it sure would be nice to have better methods of rodent control in urban areas, ideally just by suppressing their reproduction. It'd be much more humane. A lot of animal suffering caused by our using poisons against rodents and glue traps and other things. But what about Aotearoa? Should it be used there at all? So we effectively said, look, we want Maori co-governance over any proposals to use stuff that's our doing in Aotearoa, and we won't support it if Maori aren't broadly supportive. And we're interested because, well, we feel that folk who don't share Maori values and expectations regarding ecological outcomes should still take their concerns seriously, even if it can't be translated. And this is crucial because we've seen over the past few decades that there's a lot of traditional ecological engineering arrangements, really, that we've gone in and said, hey, look, this crop grows really much better, much faster. You can get an whole extra crop cycle in. You should start planting it. And it's just completely destroyed this culturally evolved natural way of managing the local ecosystem because we didn't understand how it worked. And it took us decades to figure out how it worked. And there have been some predictions in Aotearoa that practitioners of the Mātauranga traditional knowledge made that took over a decade for a Western PhD ecologist to verify, yes, indeed, that is in fact how it works. So we know it takes a long time to translate, and that's just not a feasible thing to expect. So we just have to take their word for it. But here is where I really screwed up. In the first paper describing CRISPR-based gene drive, I had to make the pretty Venn diagram showing all the possible applications. You will note controlling invasive species and pest management are on there. Now, in the main text, I noted that, since this was referred to the self-propagating form, that since rats are stowaways, and there is gene flow between all the populations pretty much everywhere in the world, if you use this on invasive rats somewhere, the gene drive will itself prove invasive and go back to the native habitat. So you can only do it if you try to do it globally. The whole world decides that you're going to get rid of all the invasives and protect the native populations. It's probably hard to coordinate internationally. So all of that is in the main text. Now, how many of you read the main text of all the papers, as opposed to looking at the figures? So a lot of people got really excited, because if you're a conservationist, you don't have a lot to be excited about of late. It feels like doom and gloom all the time. And so promising new powerful tools that could actually make a difference, you're really keen on going for it. So a lot of people got really excited about using, in particular, self-propagating CRISPR-based gene drives against invasive rodents in Aotearoa, including folks who are advising the government. So, problem is these things are super invasive. So we modeled a drive system that only copies itself half the time, and the rest of the time it makes a resistant allele that can never be copied over. So it caps out at 37% of the population-ish, and then the drive just goes down after that. But this plots the peak allele frequency reached of this very terrible gene drive against the number of organisms released and the probability density. So you can see that the probability that it will not in fact spread goes down very, very, very rapidly in the single-digit number of organisms. These things are super invasive. And if people will make money from moving something, they will move something. And we know this from Aotearoa. They smuggled in rabbit hemorrhagic fever virus, rabbit hemorrhagic disease virus, excuse me, from Australia. After that test went wrong and they spread off of the island that it was used as a test site to the mainland. And the government of New Zealand said that was not a well-run test, no importing it yet, and the farmers smuggled it in anyway. Rats cause a lot of economic damage. Someone would move it. So with Neil Gemmell of the University of Otago, I wrote a paper saying conservation needs to use localized forms of gene drive. We outlined a form of a daisy drive that splits up the element so it can't spread indefinitely, and there are various toxin, antitoxin versions, and things like the underdominance trick and that you could use, private allele targeting, various proposals. But the point is, you need to not use the self-propagating full power version. But because Neil wanted to use Aotearoa as an example, and we wrote this, and then I reached out to the Maori, and then this came back to the revision stage, and I just didn't think to send it to them, because we'd already written it, it was a revision stage, we did run it by a Pakeha scientist in Aotearoa. But turns out, when you show people in power that they are wrong and they cannot do the thing that they wanted to do and had invested some political capital into doing, and then you take that away from them, they are not happy. And it is hard to strike against me because I am here on the other side of the world. So who do they take it out against? The folks on the bottom. So as one of my Maori collaborators said, his, meaning my naivete of the political situation Maori are in publication of this paper without talking to the other partner more than likely will have consequences for a Maori that the author did not consider. And she is entirely correct. I was thinking of engaging with the Maori as drawing knowledge of their local natural ecosystems, such that nothing would go wrong. I didn't think that there's a local political ecosystem. So I apologized because I really screwed up. And they generously invited me back at a large hui on the local Tonga species and how best to preserve them. And I told this story. And the most heartbreaking comment from the audience that I've ever gotten is after I gave this talk and described the ways that it might be done and asked for their guidance, a woman, and explained that I thought I'd learned from this, but I'd already failed them. And so I would completely understand if they said, no, we don't want anything to do with this. A woman of probably 60-something rose to her feet and she said, I have never heard a Pākehā scientist apologize to us for anything before. So they did decide that they were still interested in working with us because, I mean, that's a very low bar to clear, but so we engaged in further discussions and went back numerous times pre-pandemic, brought the lead Maori coordinator at the New Zealand Environmental Protection Agency, who's in charge of integrating the Mātauranga into assessments, came out to MIT on a fellowship and joined us. That's him right there, Doug. And we continued this until the pandemic prevented us from going back. It's really hard to maintain relationships via Zoom during a pandemic. So we're hoping to raise the funds to go back and continue this, particularly in the event that our Y-linked suppression research continues. But the point of this story is that when we gain the power to act and we use it, then we become responsible for the consequences. Anything that goes wrong is on us, anything unexpected. But if we gain the power to act and we don't use it, we choose to do nothing, then we become morally responsible for the continued existence of the problem. It's on you either way as soon as you gain the power. So now we increasingly have the power to intervene in natural ecosystems in targeted and programmable ways. And it's on us no matter what we choose to do. Thank you.
Given the intimate setting of this room, we haven't got a microphone, but
this is a comment. This is the most responsible talk I've ever heard about gene engineering. And I appreciate what you're willing to stand up and say. And we as scientists need to listen very, very carefully to what you're saying, because there are all kinds of issues you just scratch the surface of. But thank you.
Thank you. There is definitely no easy answers to this one, although this isn't what keeps me up at night. Biology gets more harmful than this.
What does keep you up?
I was afraid.
You really want to know?
Yeah, I do actually.
A lot of our colleagues are very interested in learning more about pandemic viruses under the assumption that we can better prepare for them. So logically, you want to know how is something like COVID, once it jumps, going to evolve so that you can engineer vaccines that will cover the future variants. But as Richard Feynman said, what I cannot create, I do not understand. Eventually, we will understand how pandemics work. We will be able to predict antigenic evolution and cell entry of key receptors for probably all the different families of viruses. There's a lot of machine learning work on attempting to model these. And we will learn how to make arbitrary pandemics. And right now, we cannot defend against pandemics pretty much at all in any kind of effective way because of the logistics of vaccine production. In other words, many of our colleagues, very well-meaning, trying to help, are putting out their public tools for engineering pandemic-capable viruses. That's
Okay, now that's all of
We asked,
but it's totally solvable. It's just it's not solvable using our tools. It's probably not solvable within biotechnology just because you can't get the cure to everyone. You can't vaccinate everyone against everything. There are so many possible configurations of entry proteins, for example. You can't vaccinate against all of them. Even broad spectrum isn't broad enough against that kind of design capability. And if you wait until the thing is spreading, Omicron infected half of Europe and a quarter of Americans within 100 days of being discovered in southern Africa. Something spreading through airports, something deliberately released across multiple airports would spread much more quickly. And the National Biodefense Strategy says, have a vaccine approved in 100 days and have enough doses available for all Americans in 130 days. It's just way too slow. So biotechnology appears to favor offense. It's just straight up offense dominant. That doesn't mean that technology is offense dominant, because we know perfectly well how to build protective equipment that will perfectly keep us from getting infected. A powered air purifying respirator, you're not going to get infected with anything while that's on. You know, you can't scratch your nose on it, but it's also not going to break the seal if you reach under it to scratch your neck, because it's positive pressure. And those cost $1,000 each, and if we had one of those for every essential worker, then we could lock down and withstand and eliminate any pandemic. The question is, will we invest in doing that before we learn to make pandemics? So that's the other thing that I worry about these days, is that we're not thinking about the downstream consequences of just learning more. Because it's nice to believe that all knowledge is worth having, but some knowledge is not worth having yet. Not until you invest in the defenses first. Yeah.
There was a conference in Salem when the recombinant DNA started coming along. And that actually was useful, I think. I mean, my perception is that it was useful. Do you think that we could have a similar kind of moratorium occur?
Bluntly, right now, no. We did have a moratorium on enhancing transmissibility and virulence of pandemic-capable pathogens during the Obama administration, and then it was overturned. But too many of our colleagues think it's a good idea to learn which mutations might occur that would make things pandemic capable, so that we can then better try to prevent spillover, early detection, all that jazz, which is a very reasonable thing to think. It's just, it assumes there's nothing deliberate.
Yeah, you've shown that that thinking is not necessarily.
But it's very intuitive. You want to know more, because then knowing about it makes you feel safer, and then you do have more options for responding. It's just, we spend so much of our time thinking about how to fight disease that it's hard to conceive that someone might want to use it as a weapon. But it only takes one person.
Right.
Interesting. If I could, Salem State brands itself as Massachusetts Civil Engagement University. I think that's how they describe it. And we have an office here for an Office of Civic Engagement. I have done work in Southeast Asian tropical rainforest and Puerto Rican dry forest and locally. And I didn't have support for the first two, but I have support now through that office. And it's made a real difference. I have a community-based participatory research project that follows a certain structure. And I find it to be a structure that is working. It's not always smooth. But I feel like we've accomplished a lot in 4 years. Do you have that type of support at MIT? People you can go to say, this is the next step I'm going to take. Could you give me some feedback about the right path forward? Like, or I'm running into an issue. Because this is what I do. Like, if I get to a new stage of the project, I reach out to them and I say, this is where I am. What are the pitfalls? What do I need to watch out for? I'm wondering if you have something similar at MIT.
Actually, we really don't. So we have the lawyers that tell us what we can't do.
Lawyers can be useful.
And we have the committee that tells us what we need to be careful of when it comes to national security and engagements with China and so forth. We don't really have that. I mean, the Office of the Vice President for Research handles weird things like that. So we just developed some phage mid tracers to put into the sewage system at different points, just flush them down different toilets with different barcodes to then figure out how long does it take different pathogens flush down a toilet to get to Deer Island treatment plant. So just figure out how good is wastewater monitoring for pathogen detection in a city environment. But because that would involve putting something into the environment that is engineered, then this is, sort of trips the warning flags and is you need permission from the Vice President for Research's office. And so they look at it and make sure, and then they basically say, have you engaged with every relevant regulatory authority already? And I have learned that this is the thing you have to do. You have to go to them and saying, yep, here's the local, state, and federal regulatory approval, and here is a letter from the Office of Science and Technology Policy at the White House saying that they approve of this. And then maybe you'll get it through MIT. Sometimes I can't fathom how we go full throttle on understanding how pandemic viruses work, and yet we're so constrained and cautious on things like this. So no, we don't have anything specifically for supporting that. It sounds lovely, and it's amazing that you're doing it. We don't have anything quite like that. We do have the advantage of the city of Cambridge has a biosafety committee for the city, originating from the original recombinant DNA moratorium. And the city of Boston has one too. So we often talk to them. And the other resource that we do have at MIT who are really great, our environmental health services group is which runs our biosafety committee, which I'm on. And our biosafety officers are really great, and they talk to us about this, help us engage with the EPA and so on. So they're the closest equivalent, but they're not really about going out to the community, except insofar as saying, you should talk to the city biosafety committee because they're the community.
I mean, I think it would be a lot harder for them to support me if I were doing work in New Zealand, right? Like,
Right.
Because here they know, I'm doing the work in Salem, so they have personal and professional connections throughout the city. So with the community, but also with the mayor's office, for example. So those types of connections are really helpful. But the other element here is that makes the community-based participatory research, I don't mean to make this about me, sorry. But the thing that really works well is that I have partners, I'm a partner in the city and a partner in the community, and we are equals in the project. And I always consult with them before each step. And they do the same with me, and they bring different types of expertise. So, that's super, super helpful, because sometimes my community partner will put on the brakes and say,
Yeah.
No, you don't want to. I'm like, but. But I listen to her, so, and also if I have a need that from the city, and she has a way of figuring out how to get that, so it's.
That sounds very like the situation that we've established on Nantucket, where just the Board of Health Commissioner just orchestrates things, and then a former member of the Select Board handles that side of government, and then we have the local skeptic who's in tune with the people who really don't like the idea of engineering life across the board.
Skeptics are so important, right?
Mm-hmm. Absolutely.
Yeah, because even if they're wrong, they're going to influence other people.
Yep.
So it will turn the.
Yep.
I had to do some quick troubleshooting. We had an article in the Globe, and I had to watch social media because there were some of these people who are they're negative, right? So there's maybe two, but they were popping up all over the place.
Yeah, it's really hard because some folks feel very strongly about these things, and you have to ensure that everyone's voice is heard. But at the same time, you try to avoid misleading, and you have to try to avoid letting other people mislead what's going on. I mean, people call these Frankenmice. Of course they do. That's just how it is. I mean, but for the most part on the islands, people hate Lyme disease enough that they're really interested in going for it. Although the derail is always, well, how about we just shoot all the deer? It's like, you've tried that many times before. And they're like, no, but I've never been on it before. I am so charismatic and persuasive, and I will convince everyone that we will shoot all of the deer now. It's like, you go do that, and we'll be back in a year and see how that's gone.
I remember hearing about that effort on Mohegan.
Yeah.
Mm-hmm. What was the outcome there?
It mostly worked.
It
mostly worked. Yep, that's the strongest evidence that, in fact, if you just shot all the deer, then the problem would mostly go away.
That's great.
Yeah. So Sam.
We have a question.
Yeah.
I think you were the first to kind of associate the terms CRISPR and gene drive. Can you say something about how other people use CRISPR and what linking that to gene drive actually means?
Yeah, so CRISPR is normally, well, actually saying what people normally use CRISPR for. I mean, people use CRISPR for almost everything now, just because CRISPR is a protein that allows you to input an RNA, and it will then go and find some other DNA, some matching DNA sequence or RNA sequence in the cell. So you can, natively it's scissors, and so people use it to cut the DNA, which then triggers the cell to try to find a way to repair it. So if you provide the new version to be inserted, then the cell will often insert your new version to repair the damage, as though it were copying it over from the other chromosome. But you can also nick just one strand and cause other things to happen. You can attach a base editor to change just one base. You can nick one strand and then have a reverse transcriptase make a new single-stranded DNA sequence to be inserted. That's prime editing. You can break the scissors function and attach an activator or a repressor to turn genes on or off with CRISPR. But the native form is just use the scissors to cut it and then introduce new DNA sequence to be inserted. So in a mouse, that's called a knock-in. But the gene drive is just encoding the editing machinery along with the edit, such that editing can happen every generation. So that's the gene drive aspect. And gene drive occurs very commonly in nature. And that particular mechanism of an endonuclease on one chromosome, cutting the chromosome that doesn't have it so that the gene encoding that endonuclease gets copied over, that's very common in yeasts in particular, also in some bacteriophages. What's new about CRISPR is it's just an endonuclease that we can very easily target. And I was far from the first one to suggest we use gene drive to engineer wild organisms. That was Austin Burt of Imperial College London. And he actually launched a project trying to get it working in malarial mosquitoes using an actual yeast homing endonuclease. Unfortunately, as someone in the directed evolution field, you do not want to engineer homing endonucleases. They are a tremendous pain, extremely, extremely difficult to retarget. And if you only cut at one site, you run the risk of acquiring a mutation that then would block cutting thereafter. So that's the resistance that I referred to that capped out that poorly functional gene drive. What you want is to cut multiple sites. And CRISPR lets us do that very easily. And CRISPR also lets us ensure that the process is reversible, just because, and this is in fact why CRISPR-based gene drive is defense dominant, which is rare in biotech. Because it is slow, spreads only vertically over generations. It's obvious to sequencing, you can't hide it. You don't see CRISPR systems in sexually reproducing genomes. And it's easily countered because any CRISPR-based gene drive can be overwritten by another CRISPR-based gene drive that is identical except for the addition of some extra guide RNAs that targets the first one and engineers it so it doesn't target itself. So that way you can just take one that is doing something wrong, remove whatever it is about it that's doing it wrong, and then program it to overwrite the first one. And because of the dynamics of reproduction in the relevant organisms, you effectively have a defender advantage. You can always raise more mosquitoes and release them to counteract a gene drive that is doing something wrong. You can't actually turn the population to the exact genetic sequence it was before. That is beyond us. But you can ensure that the phenotype will go back to what it was before. And when we built the first examples of CRISPR-based gene drive in yeast, we simultaneously built one version that was intended to knock out a native gene. And then at the same time, we built the countering version that overwrote the first one and restored the gene. And they both worked. But we wanted to set the precedent of don't build a gene drive without building the countering immunizing reversal drive. That one everyone ignored, but we tried.
Yeah, you had a question.
Yeah, I was just wondering, with a disease like Lyme, where you're not relying on herd immunity because it's not transmitted from person to person, why is there a preference, or maybe there isn't a preference, maybe there's another reason, but why go to the vector, why engineer the vector and, instead of just working on a vaccine for humans for Lyme disease?
So so the history of the Lyme vaccine is kind of a mess. There used to be one for humans, still available for dogs, but it was withdrawn from the market because there was a backlash. It was at the very beginning of the anti-vax movement. People were claiming that the vaccine was giving them Lyme arthritis. And the FDA investigated and concluded that it was not in fact happening, but people believed it was happening, and there was campaigns and lawsuits against the pharmaceutical company that produced it. And because it had only been approved for 15 and older, and it wasn't very commonly taken up, they weren't really making money on it anyway. So then in the face of all these lawsuits and bad press, the company just said, well, enough with that, we're just gonna withdraw it. So they still sell it in the veterinary market. It's literally the same vaccine. So if you want a Lyme vaccine, you just have to have a friendly veterinarian. It's about 75% protective, which, that's better than nothing. It doesn't get rid of the problem, but it would help a lot. And thankfully, there is a new one being developed where this time the strategy was to include coverage of the European strains so they would also be effective in Europe, because the original one was only the North American strain. So they're hoping that the European market will help. So hopefully that will come out. But even that one is also still only about 75% effective. So that would be much better, especially if kids can get it. But they're probably not going to approve it for, age 1 and up. So there's going to be a window of vulnerable kids, and still 25% is still quite a lot of risk when it comes to how nasty Lyme disease can be in the chronic form. So, but the real answer is we needed some problem sufficiently obvious to try to figure out how to develop eco-technologies. And that's why we did this, not because we decided Lyme was the most important thing that we should be working on, in all honesty.
One or two more questions.
Yeah.
For Kellen. Gwen?
Yeah, is anyone there an immunologist? You saw it with immunologists at MIT?
So our primary immunologist is actually our collaborator at Dana-Farber,
Oh, I'm sorry.
Wayne
I
Marasco.
see a picture. I was
Yeah.
going to suggest that if you...
Yeah, we don't have a primary immunologist in my group. We have people with a wide variety of forms of expertise, but immunology is not one of them. Yeah.
It's fine. Can you say, what is there about the hamster that made the white-footed mouse closer to that than to other types of?
Oh yeah.
I've heard a lot of random mouse strains and all of the genetics, so.
Yeah, so it's just.
More hamster-like.
Yeah, so I was just noting that where was that? I was just noting that there we go, that they're more closely related evolutionarily to hamsters than they are to lab mice and rats. And reproductively, they're also more similar, so our activity monitoring trick actually works in all of them, but it seems to actually work better in these. And so we're making it easier to engineer hamsters as well. So if you want a glowing hamster.
Kevin, you told us a great story, a heartwarming story following your Maori faux pas, and your trips out to Nantucket and Martha's Vineyard stand back to, what was it, 2016?
Yeah.
In your group, who was the impetus to let's go out and be humble scientists to ask? Was it you personally, or was this a new movement in the guys that work in this particular field?
It was mostly me trying to figure out how to engage in this topic. And then I was fortunate enough to be joined by Joanna Buckthal, who's the project manager of Mice Against Ticks. She joined as a student in my lab not too long thereafter. And Sam Telford, who's at Tufts, is our Lyme epidemiologist and ecologist. And I invited him to just a preliminary discussion of how should we go about this? Is this a totally crazy idea before I go and talk to these communities? And Sam is amazing. So he was very much, oh wow, this is actually a novel way of tackling this recalcitrant problem. And so he was very much on board with helping. And he's just been, honestly, one of the best collaborators I've ever had. So, yeah, no, and then we were very fortunate to connect with the right people on Nantucket very early. Yeah.
Well, thanks very much for
Right.
joining us and for